Assalamu alaikum, respected ma'am. Hope so you are fine. I am Aliza Arshad and I am from Okara. I am the student of biotechnology, MPhil biotechnology, and my roll number is 5025. Today I am discuss with you the selection and the screening of recombinant transformants. First of all, the main step of the transformation is selection of transformants. In recombinant DNA technology, after introduction of recombinant DNA molecules into the host cell, it is important to select the host cell that take up the DNA construction from those that do not, in which we use two types of gene. हमारे पास यहाँ पे सिलेक्शन के लिए ट्रांसफॉर्मेंट्स की दो तरह के जीन जो हैं वो इन्वॉल्व होते हैं. द फर्स्ट वन इज selectable marker gene and the other one is reporter gene. Selectable marker gene are basically present on the vector into which the DNA of interest has been cloned. These genes protect the organisms from the selective agent that would normally kill it. All cells do not contain the foreign DNA, are killed in the presence of selective agent, and only the desired ones are left behind. There are the following common selectable markers genes are neomycin, phosphotransferase, glycophosphatase, oxidase reductase, hypoxanthine, thymine, and kinase. Other one gene is a reporter gene. An alternative or selectable marker gene is a reporter gene which help in distinguish between wanted and unwanted cells. There should be the following properties of the reporter gene. It should be easy to assay. There should be little or non-endogenic activity for this gene. It should be non-toxic and it should tolerate N-terminal fusion. We use the commonly reporter genes are chlorophenol acetyltransferase, beta-galactosidase, nopalin synthase, and octapine synthase. You can see that here the diagram. This diagram is the process of creating a recombinant plasmid and introducing it into the host cell through the transformation. In first step, you can see that here plasmid and the gene of interest. Plasmid and gene of interest, you can see that here in the circular form in this diagram, the first. Okay, next, a plasmid containing the LEU gene is combined with the gene of interest. What is LEU gene? LEU gene is basically the biosynthase that is used in the transformation. Recombinant second step is the recombinant plasmid formation. The gene of interest is inserted into the plasmid, creating a recombinant plasmid. In third step, you can see that here the transformation, the process, the recombinant plasmid is introduced into the auxotrophic host into the mutant for the gene of interest. This means that host cell cannot be produce certain substances on its own. The fourth one step is the possible outcomes are there for this after the transformation. There are the three possible outcomes. The first one is cell with self-ligated vector in which the plasmid re-ligated without incorporating the gene of interest. The second one outcome is cell with recombinant vector. The plasmid successfully incorporated the gene of interest. The third one step is the cell without vector. The plasmid does not enter the cell. These are the outcomes after the transformation. The fifth one step is selection. The cells are grown on minimal media. Minimal media contain transformants and non-transformants. What is transformants? Cells that have been taken up the plasmid either with or without the gene of interest are capable of growing or the minimal media because the plasmid provide the necessary genes like LEU2. Non-transformants cells that have not been taken up the plasmid and not capable of growing on the minimal media because they lack the necessary genes. This process is commonly used in the molecular biology to introduce the new genes into the organisms for research and or the biotechnology applications. Next one step after the selection is the identification of clone of interest. Once the transformants are selected, the next step is to identify a specific transformants containing the insert of desired gene. There are the three methods. First one is sequence dependent screening, second one is protein structure function dependent screening, and third one is complementation method. In sequence dependent screening can be achieved by exploiting the homologous sequence of desired gene. Sufficient information about the sequence of interest is required to make the suitable probes and the primers. They are the two methods for the sequence dependent screening. The first one is the nucleic acid hybridization technique and the other one is PCR technique. Nucleic acid hybridization technique, the clones of the library are transferred to a membrane made of the nitrocellulose or the nylon. You should remember जो हमारी membrane है वो nitrocellulose से मिलके बनी होनी चाहिए, nitrocellulose की होनी चाहिए और nylon. The clone on the membrane are gently lysed and DNA coming out of the cell is embolized on the membrane. The desired clone is selected by the hybridization of the embolized DNA with the labeled DNA probe. Second one technique is the PCR technique. The clone are screened by the cloning PCR method by using the primers which are designed based on the available sequence information. Here you can see that the selection and the screening, all the transformant cloning with containing the vector but not all all will contain recombinant plasmid. In first you can see that here the clone containing the vector molecules can grow. They are antibiotic resistant, reduced vector only, alkaline phosphatase. Second one is how to identify the recombinants. Blue-white selection based on the lag-Z activity. Cloning PCR predefined the restriction, digestion, hybridization, screening most common than the DNA sequence. Protein structure and the function-dependent screening. Beside the nucleic acid sequence, the structure function of its expressed product can also be analyzed. Screening the product of clone applies only to expression libraries. The clones can be identified because its product is recognized by an antibody or the ligand, or because the biological activity of the protein is preserved and can be assayed. For this method, genomic DNA or cDNA library should be constructed in the expression vector. The desired clone is identified on the basis of the structure function of the protein produced. The protein product is separated by the lysing the cell of the clones and subjecting the cell lysated or the phosphopolyacramide gel electrotransferase. The desired protein on the gel is identified with the help of the molecular markers. To confirm the identified protein, the protein bands on the gel is transferred to the nitrocellulose or the polyvinylidene membrane. This technique is called Watson blotting. The desired protein product on the membrane can be confirmed by binding it to a specific antibody and the ligand. You can see that here the master plate, addition of the radioactive label probe, impression of the nitrocellulose filter paper on the master plate, the DNA hybridization with radioactive probe, RNA probe. Treatment with the detergent, treatment with NaOH, baking at 80 degree Celsius, UV exposure, auto radiography, gene of interest. You can see that here the cloning hybridization. The third one method is complementation method. All the clones of the library are pulled into the recombinant vector. Molecules are isolated from the pulled cell. A mutant of the organism is obtained in which the gene of interest is mutated. The mutated cells are transferred with the recombinant vector molecules isolated above. The mutated cell in which the restoration of the wild type function occur are selected. Recombinant vector molecules isolated from these cells containing the wild type copy of the gene of interest. In some cases, the library can be directly conjugated with the mutant cell. The bacterial cell involved in biosynthesis of amino acid, nucleotide bases, and vitamins can easily be cloned by this method. The nucleation gene of rhizobium are were cloned by this approach. The complementation method can be used for those genes for which the restore function can easily select it. Nucleic acid blotting and hybridization. The membrane used are the nitrocellulose membrane, nylon membrane, and polyvinylidene difluoride. You can see that here the transfer clonase of nitrocellulose membrane denature, deneutralize, and cross-link DNA to the membrane pre-hybridized membrane. Label prop DNA by random priming, remove unincorporated nucleotides, add salmon sperm DNA and denature. Hybridize probe to membrane, low strategy, washes, high estrogen washes and detect the DNA probe. Transfer of nucleic acid from gel to the membrane, upward capillary transfer, downward capillary transfer, simultaneous transfer to membrane, electrophoretic transfer, and last one is vacuum transfer. You can see that here the blotting process containing the solution that contain the anode and the cathode. You can see that here the plastic supports in zigzag line and the pads, that is filter paper, is in the center of the gel. The nitrocellulose membrane, you can see that here the positive sign look like this, called the electroplating or the electroblotting. Method of embolization after the transfer of nucleic acid from the gel to the membrane or the directly supporting on the membrane. Following two methods of the embolization are used for the fixation of DNA or RNA on the membrane. The first one is baking and the second one is cross-linked by UV irradiation. Thank you so much. That's all about my topic. This is the selection and the transformants detection. Thank you so much.